All right, guys, welcome back. So today we're going to pick up really just talking about the basics of how cell signaling works in general in a multitude of organisms. So first and foremost, this pathway, this signal transduction pathway we're going to start talking about today, is actually discovered by a gentleman named Sutherland, and he happened to discover it while he was studying epinephrine's effects on cells. If you know anything about epinephrine, it is part of your fight or flight response. So, what he ended up discovering and how we now recognize how these signals get transmitted and how their responses get produced in cells is that cells really get received and processed through three major steps. You've got your reception, which is what we're going to focus on first. Just how does that signal come in? This transduction, which is the actual pathway of things that are happening within the cytoplasm, and then ultimately with the response, whatever that may be. So when we talk about reception, think about it literally like a signal coming into your cell phone, or if you've got a satellite dish on your house for TV. All right, that reception has to come from somewhere. That signal has to come from somewhere. So when we talk about this in your cells, that signaling molecule is coming from somewhere. If we're talking about some sort of local regulator, like a growth factor, for example, that can be a signaling molecule. It can be a hormone. It can be a neurotransmitter. So when we look at these signaling molecules in general, we call them these ligands. Okay, and so those ligands come in and they are specifically shaped just like a substrate to an enzyme fitting together in that active site. Those ligands bind to receptors, and these receptors are protein molecules that sit in the cell membrane, and their receiving point usually is located on the outside of the cell. So here's the inside, your cytoplasm, your extracellular fluid, or your external environment, and that signaling molecule comes floating in here, finds its corresponding receptor, so they'll match in terms of a corresponding shape. And once that receptor is bound to its signaling molecule, oftentimes that receptor then will change its shape. So that conformational change will then initiate the initial transduction that will then send a spiraling signal down through the cytoplasm until we ultimately achieve that response at the end. So there's a handful of different types of membrane receptors, and where they're located and how they function usually has to do with whether or not that signaling molecule, that ligand, is in fact lipid soluble or water soluble. So for all of your hydrophobic or your water soluble molecules, remember these are the guys that are not going to be able to directly interact with fatty acid tails in your cell membrane. And so as a result, they can't just easily cross the cell membrane and create some sort of response in your cell. They've got to get through that cell membrane first. And so what they will do is attached to receptors embedded in the cell membrane, because that protein then will cross that cell membrane and be able to initiate the response internal to the cell, even though the signaling molecule, that ligand, likely never actually crosses the membrane itself. So, when we look at your different types of cell membrane receptors, one potential option are these things called G coupled or G protein coupled receptors. So, what do they look like? Well, they've got a couple of components here. So, if I take a look at the figure, here is this G protein coupled receptor. They've got this inactive G protein that sits down here, and then you usually have some sort of enzyme also as part of this system. So these G proteins act as on-off switches. So long as a GDP, so very similar to ATP, right, energy molecule, but it's a guanine instead of an adenine. Assuming that that GDP is bound, so a guanine with two phosphates attached, that G protein remains inactive. So it's not doing anything, it's not receiving a signal, it's not sending a signal. But when we convert that GDP to GTP, right, we give it some energy, that will then activate that particular molecule. Well, how do we get that conversion to occur? I've got to attach a signaling molecule. That ligand has to come in that's going to activate this receptor, which then causes that GTP activation to this particular G protein. We activate that G protein, and it's sort of like throwing a slingshot, and sending that stone, if you will, downstream somewhere. So when that activated G protein hits this enzyme, it now activates the enzyme. In that activation process, that will then cause a transduction pathway to spark, and in the same process, will then pull off that phosphate, recreate that GDP, and return that G protein to its inactive form, until another signaling molecule attaches, and then this whole pathway starts over again. Another option are these proteins called receptor tyrosine kinases. So once again, these guys generally are existing in inactive form in your cell membrane, but what they'll do is there are a bunch of tyrosines on the sides here, okay? They've got ligand binding sites at the top, and they usually work in pairs. So what you end up needing in order to activate these guys is not just a single ligand, but instead you need a signaling molecule into both of these individual pieces. Once you do that, these monomers, if you will, once you do that, they'll create a dimer, they'll merge together. And then with a bunch of ATP and phosphorylation processes, will fully activate these receptors, and then they can then attach inactive relay proteins in your cells. And the cool thing about these guys is that they will often trigger multiple signal transduction pathways all at once. And so there's some benefits to this because I can get multiple different signals in my cells from a single molecule, right? A single ligand attaches, a single type of ligand, I should say, attaches, activates these guys, and rather than creating one signaling molecule to one cellular response, I can set off one signal molecule to elicit multiple responses in that cell, or I can amplify it. So rather than just being a sort of low-lying, maybe background signal, and response as a result, I can upregulate it. I can get a much stronger, more intense result, by being able to set off multiple pathways at once as opposed to one at a time. Then you've also got these ligand-gated ion channels. So these guys also sit in your cell membrane, okay? But they are sort of like closed gates. So you need the key to open them, and the key to open them are the ligands binding to these proteins, which is what you're seeing here. You often see these in sodium channels and calcium channels, ion channels in general. So you'll typically have high concentrations on one side, low on the other, and your cells need those ion concentrations to be present, in order to set up voltage differences on opposite sides of that cell membrane. So they'll stay closed, until we have a signaling ligand attached to that gate. It's kind of like turning the key, and now I can open the gate, and then your passive transport will occur where we've got that transfer of ions or whatever the molecule happens to be from high to low, and in that movement of that ion shift, that voltage shift, that charge shift, you'll end up creating some sort of cellular response. Once the response is created, then that signaling ligand releases and the gate closes again and resets the system. So that's all for your water solubles. Now, when we talk about lipid soluble signals, Then their receptors are usually intracellular, meaning that they're not sitting in the cell membrane, but instead they're sitting internal to the cell itself. And you usually see this with things like hormones. So the receptor protein, rather than it being, again, in the cell membrane, it's sitting inside. So that hormone, because it's lipid soluble, can go straight through the plasma membrane without any issue, attached to its receptor, and then that hormone receptor complex then elicits some sort of response. And oftentimes that response is that receptor complex actually acts as a transcription factor. So we'll turn on transcription by creating that messenger RNA. Remember, transcription factors are important for allowing your RNA polymerase to bind to that promoter region more effectively. So these complexes will then turn on specific genes, create the proteins that we need, and then cause a